My dear students, welcome to my today's online class on the next topic in the phylum Protozoa. The next topic happens to be the locomotion in protozoans. You have to study about various types of locomotory organelle. In that, you will be concentrating on pseudopodia, cilia and flagella, okay, 3 types. So, by now you know I am Dr. Meera BK, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bangalore 1. You can see my cell phone number and email ID on the slide. After the class, any queries, don't hesitate to contact me. Now, you can see few structures here. They are all different types of pseudopodia. One particular type of locomotory organelle seen in protozoans, which you are going to study in detail in next coming few moments. Then you can see the structure of flagellum and cilium, which also you are going to study. Then you will study about different types of flagella. With this background, let us start our class now. Okay? Now, what is locomotion? Locomotion is the movement of animals from place to place. Okay? Movement of animals from place to place. Why they should move? Normally, animals move in search of food, in search of mate, that is the opposite sex, for copulatory purposes, and in search of shelter or to escape from predators like this. So, there are various reasons for animals to perform this act of locomotion, which is basically movement from place to place. This is influenced by various stimuli, both external stimuli as well as internal stimuli. Okay? As far as protozoans are concerned, they show great adaptability as far as their locomotory patterns are concerned, which you are going to study in detail. In fact, you can see the lowest locomotion like amoeboid movement as you seen in amoebae, and fastest locomotory movement like ciliary locomotion, which is seen in the class Ciliata, exemplified by paramecium. In protozoans, as I was telling you, locomotion is brought about by different organelle. One such is cellular extensions. Please note, the entire body is nothing but a single cell. So, cellular extensions like pseudopodia, which are formed in amoeba. Then, pellicular contractile structures like myonims, which you see in euglena and sporozoans. Of course, this is not there in your syllabus and you are not studying in detail, but you should know, apart from what you are studying, there are other locomotory structures also. Then locomotory organelles like flagella, as you see in paramecium, flagella as you see in euglena, and cilia as you see in paramecium. Sorry for the small mistake here. Flagella are the locomotory structures in euglena and cilia are the locomotory structures in paramecium. Just exchange the terms or the places. Okay? Now, with this background, let us study the pseudopodia or cellular extension or false feet, as they are called by various names in detail. Pseudo means false, poda means foot. Okay. So they are actually called false feet. Basically, since they are cellular extensions, they are also called cellular extension. Okay. Pseudopodia or cellular extension or false feet. Okay. These are temporary outgrowths of the cell formed by some protozoans, some parts of the body, okay, that happen by the movement of cytoplasm. Then there are different types of pseudopodia. Depending on the number of pseudopodia formed on the surface, pseudopodia can be categorized into polypodia and monopodia. Poly means many, poda means foot. Several pseudopodia are formed on the surface of the body, as you see in case of amoeba proteus. Amoeba generic name, proteus specific name. So if you see several pseudopodia being formed, then you use the term polypodia. On the other hand, if there is formation of single pseudopodium, okay, single pseudopodium on the surface of the body, as exemplified by Entamoeba histolytica, a parasitic protozoan, then you use the term monopodia. Mono means single. Okay. So depending on the number of pseudopodia formed, you can classify them as polypodia or monopodia, right? Then, depending on the structure of pseudopodia, they can be classified into different types. Let us study them one by one. First one is lobopodia. As the name itself says, these are lobe-like and blunt structures formed with broad and round ends. All right. So, if the locomotory organelles temporarily are formed, taking the form of a lobe, a blunt structure with broad and round end, then you call them as lobopodia. Okay. They are composed of both ectoplasm and endoplasm. Okay. Now I should tell you one thing. See, inside the cell, the entire body is a single cell. Inside the cell you find protoplasm and nucleus. And the protoplasm present in the cortical region, cortex, outer surface, towards the outer end, is towards the outer rim is called ectoplasm. Ecto means outside, and the protoplasm which is present inside the cell, much below the peripheral region, is called endoplasm. Endo means inside. Okay. So here the lobopodia are composed of both endoplasm as well as ectoplasm. Please note. Lobopodia actually move by pressure flow mechanism. Pressure flow mechanism. There is another topic in the same chapter like modes of locomotion. There I will tell you about this in detail. Right now you can just know that they move by pressure flow mechanism, and the lobopodia are found in protozoans like amoeba proteus, entamoeba histolytica, etc. You can see formation of so many lobopods, right? This is how it looks like. Then the second type of pseudopodia is phylopodia. Phyli means thread. These are slender filamentous pseudopodia tapering from base to the tip. Okay. Then sometimes they may branch out, but seldom anastomose. There are two terms here. What do you mean by them? Seldom anastomose. Anastomosing structure, whenever in biology we refer to a reticulate structure, network like structure, we use the term anastomosing type. Okay, so they branch out, but they never form a reticular structure or a network like structure. That's what I meant by the statement that sometimes they do branch out but never anastomose. Seldom is a term used in the sense of never. They never anastomose. They never form network like structure. Okay. They are composed of only ectoplasm, unlike Lobopodia. Please note, they are composed of only ectoplasm, unlike Lobopodia, and Phylopodia are formed in protozoans like Euglypha, Lecythium, etc. Okay. Now, this is the diagram of Euglypha. You can see the branched out Phylopodia at the bottom, right? And you can also observe that they have not formed a network, all right. Then the third type of pseudopodia happens to be reticulopodia. Reticulopodia, reticulum means reticulum, that is network like. The name itself suggests what it could be. Reticulopodia are also called rhizopodia or mixopodia. Okay, reticulopodia are also called rhizopodia or mixopodia. You can see the structure of Elphidium or Polystomella, where you can see the pseudopodia in the form of threads, and they have formed a network. They have anastomosed, unlike Phylopodia. Okay, so reticulopodia are predominantly seen in foraminiferans, that is shell bearing protozoans. There is an order called Foraminifera, and to which belong Elphidium, Globigerina, etc. And these protozoans exhibit pseudopodia in the form of reticulopodia. Okay. Now, they are filamentous, profusely interconnected and branched to form a sort of network, that is anastomosing structure. And please note, the primary function is ingestion of food, okay, and the secondary function is locomotion. Sometimes the functions are taken over by, locomotory function is taken over by the other function. Here the primary function is ingestion of food rather than locomotion, and executing locomotory function becomes secondary. Okay, and exhibit two-way flow of the cytoplasm. They exhibit two-way flow of the cytoplasm. All right, this is about reticulopodia. Then there is one more type of pseudopodium being formed called axopodia. You can see the diagram of a protozoan, right, actinophores, which exhibit the formation of axopodia. Okay, so axopodia are formed in protozoans like actinosphaerium, actinophores, colosome, etc. The diagram what you are seeing on the slide is actinophores. Okay, now let us see the details. Axopodia are fine needle-like straight pseudopodia radiating from the surface of the body. Fine needle-like straight pseudopodia radiating from the surface of the body, as you can see in the diagram. They contain, each axopodium contains a central axial rod covered by granular and adhesive cytoplasm. Central axial rod covered by granular and adhesive cytoplasm. Okay. Then the main function of axopodia is, again please note, food collection rather than locomotion. These axopodia also exhibit two-way flow cytoplasm like the previous one. Okay. Then axopodia are mainly found in heliozoans and radiolarians. Heliozoa and Radiolaria are two orders under the phylum Protozoa, and protozoans which belong to these orders normally predominantly exhibit oxopodia. Okay. So, till now you have learnt the pseudopodia, different types of pseudopodia with examples and their functions. Okay. Now, as I was telling you, please note pseudopodia, cilia and flagella are there in your syllabus to study the locomotory organs of organelle of the phylum Protozoa. But please note, other than these three structures, you do find few structures like pellicular contractile extensions in the form of myonems in protozoans, which do serve the purpose of locomotion. Okay. So, they are seen in Euglena, a flagellate, then in larger ciliates and trypanosoma in the form of ridges or grooves or contractile myofibrils or microtubules. Okay. So, these are also locomotory organelle seen in different protozoa, but in-depth study is right now not needed, as this is not there according to your syllabus. Treat this slide as an additional or supplementary information. If you are interested, you can go to any textbooks and make a detailed note of it, or come to me whenever I am free when you come for contact classes. Okay. I will just skip this. Then let us study about flagella as locomotory organs. Locomotory organs of flagellate mastigophorans protozoans are present in the form of flagella. Okay. So, in Mastigophora or class Flagellata, to which belong Chlamydomonas, Euglena, etc., there you find flagellum singular, flagella plural as the locomotory organelle. Then they appear in the form of thread-like projections on the cell surface. They appear in the form of thread-like projections on the cell surface. A typical flagellum consists of an elongated, stiff fiber called axial filament or the axoneme. Please do remember the terms. Lot of 1-mark questions can be asked in such places. Okay. So, a typical flagellum consists of an elongated stiff fiber called axial filament or axoneme, which is enclosed by a protoplasmic outer sheath called axoplasmic sheath. Okay. Then the axoneme or the axis arises from a basal granule, a granular structure called basal granule, and this basal granule often in protozoans is referred to as blepharoplast or kinetosome, blepharoplast or kinetosome. And please note, basically the kinetosome or blepharoplast is a product of centriole. You must be knowing that in an animal cell you find centrioles, in plant cell you find centrosomes, right. So, these centrioles are the ones which give rise to the basal granule called blepharoplast or kinetosome, from which a typical flagellum arises. Okay. So, this lies below the cell surface in the ectoplasm, that is outer protoplasmic area. The region around this basal granule or blepharoplast is referred to microtubular organizing center, microtubular organizing center that controls basically the assembly of microtubules. Please note, students, when mitosis is taking place, centrioles do the same job, that is regulating the assembly of spindle fibers, right. So, same function but in a different context, alright. This is how you have to correlate the things in biology and analyze for your cells. Yeah. Here you can see a diagram. The first one shows the longitudinal section of the flagellum, and when a cut is made, when a section is taken, how it looks like is the blue color one, and below that you find the cut section of cilium. Okay. Now let us study how a flagellum can be described or how a flagellum looks like, and if you understand the structure of a flagellum, it is very easy to understand the structure of a cilium, because essentially their structure remains almost similar, okay, but for here and there minor differences. Okay. Now with this background, let us study the structure of a flagellum. So I told you that in the center you find an axial filament or axoneme. This, if you look under an electron microscope, shows typical 9+2 arrangement. You can just see the cross section here. In the center you can see two bodies, and outer side you can see, just count the number, they are 9, right? 9, and each unit is made up of 2 subunits, therefore we call them doublets, unit A, subunit A and subunit B. Okay. I think if you carefully observe the cut section of flagellum, you can see this 9+2 arrangement right in the diagram, right? The two central longitudinal fibers, okay, please note they are not attached, they are separate. Why? I will tell you in my next class. The two central longitudinal fibers are enclosed by membranous inner sheath, the green one, what you can see in the first diagram, right? So in the center you find two central longitudinal fibers, and they are enclosed by membranous inner sheath. Then the two central longitudinal fibers are surrounded by 9 longitudinal peripheral doublets. You can count them, they are exactly 9, and each unit has 2 subunits, therefore you use the term doublets. In cilia, at some places you find triplets instead of doublets. That is one little difference between this, you can see in the third diagram. Okay. Now the two central longitudinal fibers are surrounded by 9 longitudinal peripheral doublets, and each doublet is made up of 2 microtubules, microtubule A and microtubule B, which are in the form of a cylinder. In the first diagram you can make out, which are in the form of a cylinder between the inner and the outer sheath. I repeat, don't get confused. The axial filament, which is the stiff central structure of a flagellum, under an electron microscope shows typical 9+2 arrangement. What it is? The two central longitudinal fibers are enclosed by membranous sheath, and these two longitudinal fibers are surrounded by 9 longitudinal peripheral doublets made up of 2 microtubular units A and B. All right, which form a cylinder between the inner and the outer sheath. All right. Then each peripheral pair, if you carefully observe, each peripheral paired fiber is connected, okay, by radial spokes. They are connected to the internal membranous sheath by what are known as radial spokes. Let me come back to the previous. You can see the blue colored dots running from the doublets to the central longitudinal fibers, right? They are called radial spokes. Okay. Okay. Then each peripheral doublet has pairs of projections directed towards neighboring doublet. All right, and these projections are arms, are made up of protein called dynein. They are made up of the protein dynein. Right? One more question. Please remember. Then, these arms, what do? They create sliding force. The arms create sliding force. Okay? The peripheral doublets are surrounded by an outer membranous sheath called protoplasmic sheath, which is nothing but an extension of which is an extension of plasma membrane. Now, some flagella, this is the basic structure. Other than this, some flagella also bear lateral appendages called flimmers or mastigonemes. Okay? Lateral appendages called flimmers or mastigonemes along the length of the axoneme above the level of pellicle. Okay? When I show you the diagrams, it will be very clear. Let me show you. Based on that, you can classify the flagella into different types. Okay? So, before going into the types of flagella based on the arrangement of lateral appendages, let us see the types of flagella based on the number. Okay? So, free living flagellate species like Chlamydomonas, Euglena, etc., normally they will have 1 to 8, 1 flagellum to 8 flagella. But the parasitic forms, may have 1 to many flagella, right? So, the number of flagella varies in different mastigophora from 1 to 8 or even more if it is a parasite. Then, flagella are classified based on the arrangement of lateral appendages and the nature of axial filament. This now let us study. One type is stichonematic. Stichonematic, what happens here? Only one row of lateral appendages occurs on the axoneme, right from bottom to the tip. This is seen in protozoans like Euglena, Astasia, etc. I will show the diagram in the next slide. You can see here the first one. You can see the flagella, you can see the lateral extensions, right? Lateral extensions only on the one side, right? Then, second type is pantonymatic, the second one, where you can see 2 or more rows of lateral appendages. You can see on either side of the axoneme, right? Lateral appendages occur on the axoneme. So, pantonymatic type is seen in protozoans like Paranema, Monas. Dear students, it may be little difficult for you to remember all the terms and examples, but there is no other alternative. Daily you have to read and practice them once, because these are all the places where the paper setter will catch 1 mark questions. Okay? Don't ignore. Okay? Then the third type is acronematic. Lateral appendages are absent. You can see in the third diagram, right? And axoneme ends as a terminal naked axial filament. You can see thread-like structure at the tip, right? So, axoneme ends as a terminal naked axial filament. So, this type is seen in protozoans like Chlamydomonas, Polytoma, etc. Alright. Then, third type, pantacronematic. Okay, pantacronematic. Here, this is fourth diagram. Flagellum is provided with 2 or more rows of lateral appendages, and at the same time, the axoneme ends in a terminal naked axial filament. Okay, so this is seen in protozoans like Arceolus. Alright, so in the first type, lateral extensions on only one side. In the second type, on 2 sides. In the third type, no lateral appendages, and the axial filament ends as terminal tip, right? Naked axial filament, right? And then in the fourth type, what happens? Two or more rows of lateral appendages and axoneme ends as a terminal naked axial filament. Okay, don't get confused, but you need to know all these different types of flagella. Okay. Then one is anematic, as the name itself says. In some cases, a simple flagellum is seen without any lateral appendages or any terminal axial filament. This type of flagellum is called anematic flagellum. This is seen in protozoans like Chylomonas, Cryptomonas, etc. So, this is all about the ultrastructure of flagellum. Now, let us study the cilia as locomotory organelles. You can't use the term organs, it is for a multicellular animal. You have to use the term organelle. Okay, cilia as locomotory organelle. Let's see. These are short hair-like structures present all over the surface of the body. You must have seen the diagram of a paramecium in my last class, right? Sometimes they may also be confined to specific region of a particular ciliate protozoan. Say for example, in Verticilla, the cilia are confined to the vestibular region, that is mouth region, right? Whereas in paramecium, the cilia are distributed all over the surface of the body. So, they may be seen all over the surface or they may be confined to the specific region. Okay. Then what do? They do primarily help in locomotion and secondarily help in food collection. This food collection function you will know in detail if you happen to study the cytopharyngeal region of paramecium in detail, and unfortunately it is not there in the syllabus, but you can give a second reading, alright. Then, yeah, cilia greatly resemble the flagella in their structure. I have told you already. The major difference between flagella and cilia is that cilia are smaller when compared to the flagella. Cilia also arise from kinetosome or blepharoplast or basal granules, which takes its origin from centriole, same. Then, cilia consists of an axial filament called axoneme surrounded by a protoplasmic outer sheath, similar to flagellum. If you happen to study the electronic macroscopic studies of an cilia, axoneme typically reveals 9+2 organization, right, similar to the flagellum. So, 9+2 organization of peripheral doublet fibrils and central singlet fibrils. Okay. Then, the details of 9+2 organization and the presence of dynein arms, that is dynein protein arms, are similar to that of flagellum. Therefore, I told you, if you understand the structure of flagellum correctly, it is as good as understanding the structure of cilia. All these fibrils are embedded in a matrix in both the cases, and the central fibrils are enclosed in a delicate sheath. Now, yes, this slide intentionally I have put here. See, if you happen to study neuromotor system or nervous system, especially in the cytopharynx region of paramecium, it is quite complicated, revealing infraciliary system, all right. How exactly the system functions, but the details of this is beyond the scope of your syllabus. Here I have given this as a supplementary information. If you are interested, you can just go through some books, and any doubts you can contact me. This is too much for your level, okay. Therefore, I just skip it. Then types of cilia. In some primitive forms like Holotrichs, that is order Holotricha, to which belongs paramecium, Cilia are present all over the surface of the body, whereas in some forms like Peritrix, example Verticilla, cilia are present only in the peristomial region. But please note, a very tricky situation may also arise wherein insectorians, to which belongs the protozoan Acinata, please note cilia are present only in the young ones, and later they are replaced by sucking tentacles in the adults, okay, according to its mode of nutrition. All right. So these deviations may do happen in biology, okay, which you need to know and make a note of it. Now, let me tell you the differences between the flagella and cilia. Flagella, I have told you, maybe 1 to 4 in number, maximum 8, more than 8 in case of parasitic mastigophorans. Whereas cilia are present in enormous numbers, anywhere from 3000 to 14,000 in number, and they are quite small. A flagellum measures about 150 microns in length. Micron, you mean 10 to the power of -6. By now you must be knowing this. And a cilium is about 5 to 10 microns. Just compare. Okay. Then flagella are commonly found at one end of the cell, normally in the anterior end of the cell, whereas cilia occur either all over the body surface or at particular specific regions of the cell, as I have already explained you. Then flagella produce undular movements. I will tell you about this when I take up modes of locomotion in protozoa, and cilia produce cilia produce peripheral movements. Okay. Then flagella help in locomotion only locomotion. Cilia help in feeding and locomotion. All right. Flagella do not form compound organelle. Cilia may form undulation membranes and other compound ciliary organelles. Dear students, till now I have tried to present before you a brief account of different types of locomotory organelle that you see in protozoans. Okay. Thank you for your patient listening. Any queries you can contact me. Till then,